In this video, we will see what is gateway cloning and how it is different from classic restriction enzyme-based cloning. I will go over some basic knowledge and link it to simple gateway cloning strategy as well as complex gateway methods. I will discuss specifics of LR and BP reactions and dive into their steps in a bit of detail. Finally, we will see what makes gateway great and some reasons to stay away from it. Gateway cloning has been around for more than 2 decades and is commercialized by Invitrogen. This means you can buy it as a kit. It is a method to clone without using restriction enzymes. Traditionally, in restriction-based cloning, you start with a vector, a plasmid backbone, into which you would like to insert something. So you digest and perhaps purify the vector. In parallel, you prepare the insert, which could be a PCR product or perhaps a piece of DNA from another vector, which means you are subcloning. Depending on your strategy, you digest, maybe you blunt, and purify the insert and combine it with the backbone. This ligation gets transformed, selected, screened, and eventually validated, possibly for orientation if you do blunt cloning. I have a separate video that discusses these things in much more detail. These steps from start to the ligation are labor-intensive. Gateway cloning replaces all these upstream manipulations and ligation steps in one super quick reaction. So what is happening in this? Gateway cloning relies on at-site recombination. At-sites are attachment sites and they are DNA sequences. Here's some context to attachment sites. Think of a happy bacteria that is swimming, but out of nowhere a bacteriophage lambda attacks the bacteria. The infection leads to viral DNA injection into the bacteria. If you know a little bit about the virus life cycle, the viral DNA can integrate into the bacterial genome. This integration occurs due to recombination of the attachment sites between the two genomes. So both virus and bacteria DNA carry at sites. Phage at sites are called attP, bacterial at sites are called attB. Depending on how you define them, they range from 25 to 300 nucleotides in length. A typical at site is split into 2 portions, say P and P', and the middle recombination spot is called O. Likewise, you have B and B', and the middle spot is called O. The O is the recombination spot between attP and attB. Let's actually understand how this viral integration works and how at-site recombination is involved in this. The virus DNA, as we said, has the attP site. I'm drawing the virus DNA as circular for ease of understanding. Instead of calling these 2 halves of attP as P and P', let's name them 1 and 2. The attB of bacterial DNA likewise has B and B', but let's call them 3 and 4. When the viral DNA needs to integrate into the host genome, it makes an enzyme called integrase. The integrases recognize attB and attP and cause a recombination to occur between them. This recombination between the 2 at sites is called a BP reaction. Recombination event occurs at the O region between the 2 at sites. If you start from the perspective of site 3, the recombination brings number 2 in front of 3. And now this entire viral DNA until number 1 comes along with number 2. But now the number 1 gets attached to number 4. From number 4, we get back to the bacterial DNA, which comes full circle to number 3. From this BP reaction, we see that the virus DNA has integrated into the bacterial DNA. And that is the prophage life cycle of the virus. Also, just to get back to these chimeric BP sites, this BP site is called attL, and this PB site is called attR. These stand for left and right. You may come across some old literature that calls them BOP or POB, but I will use attL and attR terminology. Now that we understand this, let's look at a simple gateway cloning. This uses 2 attP and 2 attB sites. Assume that our final vector will be this blue vector carrying a kanamycin marker. It carries 2 attP sites with a CCDB suicide gene in the middle. The second vector carries 2 attB sites, and it has the DNA of interest we want to clone in the middle. This is a donor vector. Just like this DNA of interest being in the plasmid form, you can also have the DNA of interest from a PCR, but in that case you will have to add attB sites to the primer. For simplicity, I will restrict the discussion to the plasmid form of the donor vector. For gateway cloning, you take these two plasmids and combine them with integrases in a BP reaction. This causes recombination between the attP and attB sites. So now we can redraw the final vector, and let's start with this part of attP, which gets switched with attB. And then the DNA continues until you have half of attB, which gets recombined with attP. And this comes back full circle. Essentially, we have swapped DNA into the final vector. Like this, you can also redraw the donor vector, which instead of DNA now contains the CCDB gene. And like we saw earlier, you can annotate these new chimeric sites as attR and attL. Note that all of this happens in vitro, and following this reaction, it is ready to be transformed, and the bacteria is grown in kinamycin because the final vector containing our DNA has kinamycin, which means any bacteria carrying this other plasmid will die. So, in this simple gateway cloning, we have learned about one simple rule. attP and attB can recombine, but attP and attP do not recombine. Likewise, attB and another attB cannot recombine. If these two were true, then bacterial and viral DNA during infection process will recombine by themselves, and that will mess up everything. Alright, so we have switched this internal portion between the two plasmids, and this is a recombination mediated exchange. But we saw that during infection, the viral DNA integrates into the host genome. But in gateway cloning, we talk about exchange. So where is the integration event in this gateway process? Let's break this process down and understand stepwise how integration can lead to exchange. We have two attP and two attBs. For simplicity, let's label the halves as 1, 2, 3, 4, 5, and 6, and 7, and 8. And to make this simple, imagine that this pair of att gets recombined first. Also, let's assume that the second pair is not used until the first reaction is complete. And we will focus on the final vector and anchor at this site 1. As we saw before, anchor site 1 recombines with site 6. Site 6 continues into site 7 and site 8. If we continue, we see this entire plasmid comes back to site 5. Site 5 gets recombined with site 2, and this site 2 continues to site 3 and 4, and all this circle backs to kinamycin. This is an intermediate plasmid where only one BP reaction has occurred, and we have integrated the donor vector into the final vector. In this, we have one attR, one attL, one attB, and one attP. The attB and P can further undergo a second BP reaction. To understand this next step, it is best to redraw the plasmid in a different configuration. This is done such that the B and P are next to each other. You can pause this video and try to draw this yourself as well. In the second BP reaction, the site 7 gets recombined with site 4, which comes back full circle to kinamycin. This releases our DNA of interest in the kinamycin plasmid. The second recombination occurs from site 3 to site 8, which circle backs to ampicillin, with the chimeric site 5 and 2 and the CCDB gene. You can label all these sites as attR and attL. If you look closely, these are exactly the same product we had originally produced. So this way, sequential BP reactions can integrate and exchange the DNA with CCDB in the donor and the final vector. Okay, now we can move to the second part of the simple gateway cloning. You remember this prophage structure we discussed a few minutes ago, where you get the full attL and attR. Another aspect of the virus life cycle is that prophage can move to a lytic cycle, where it is possible that the viral genome is excised out of the bacterial genome. How does that happen? To understand that, we have to redraw the configuration of this genome in the following way. During lytic phase, some excision integrases and other enzymes are expressed. These enzymes can recombine attR and attL. Looking at site 3, it will recombine with 4, and this gives you the bacterial DNA. Likewise, number 1 recombines with number 2, which releases the viral DNA. Previously, we have noted BP reaction that occurs between attB and attP. This excision occurs between attL and attR, so we call this an LR reaction. From the LR reaction, we can recover the attP and attB sites. Just like B and P, we can have another variant in our simple gateway cloning, where we use 2 at R and 2 at L sites. In this style of gateway, the final vector has at L sites, for example. And likewise, the donor vector will have compatible at R sites. And these two vectors will use another set of specific enzymes, and this will be an LR reaction. And the recombination occurs in a similar fashion, except that the final forms have attB and attP in the products. This is yet another example of recombination-mediated exchange. The rules for LR reactions are that L and R can recombine, but two L's cannot, and likewise two at R's cannot. The interesting bit to note is that the attB containing DNA I in the final vector can become a new donor for a new BP reaction if you wanted to use it. Now that you understand the simple gateway cloning, let me give you a snapshot of some complex ways to do gateway cloning. This is called a multi-site gateway. Simple gateway has attB and attP compatibility. However, there are variants of attB and Ps. For instance, you have attB1 and attP1, which are compatible with each other, but B and P1 are not compatible, and likewise B1 and P are not compatible. Just like B1 and P1, you also have other variants on B and Ps. Suffice to say that their products are likewise also variants, and one variant pair is not compatible with another variant pair. Let's do this with an example where you're trying to clone a promoter and an ORF into a plasmid at the same time. Let's assume that the final vector has at R1 and at R2. Then you have the two donors, one containing the promoter between at L1 and at R3, and the second donor containing the ORF between L2 and L3. In the multi-site gateway, you mix all these three vectors in one reaction, and cross-vector recombination can occur in the presence of specific LR reaction enzymes. Each variant gets recombined independently. If you solve this puzzle step by step, you will note that the promoter is flanked by B1 and B3. The ORF is flanked by B3 and B2. And this entire configuration is present in the final kanamycin vector. If you want step-by-step details on this reaction, it is available on my Patreon along with the reference material used to make this video. This multi-site gateway doesn't just stop here. You can come in with a new vector containing P1, P2, and P3. And switch out the ORF if you wanted to, or even the promoter using a donor vector containing P1 and P3. This brings us to the last segment of this video. The advantage of gateway is that it saves time, but this comes at a very high cost. But it is quite efficient. It reaches very high throughput, and it is very flexible, as we saw from the multi-site gateway. The process is virtually scarless since no restriction enzymes are involved. Multi-site gateway is also very powerful because it saves you sequential cloning steps if you were to use classic enzyme-based cloning methods. The disadvantage is that restriction enzymes are not involved. So you're actually stuck in this gateway cloning ecosystem, and the cost of switching out is very high. We did say that the lack of restriction enzymes leaves no scar, but you still have to clone your DNA into a donor vector with at sites. So it is not 100% enzyme free. This becomes very complicated if you were starting from scratch, and you have to build multiple donor vectors containing these complex sites. So restriction enzymes are still required in the beginning stages of the gateway cloning. There's one concern with gateway expression vectors, like the one here containing a promoter and an ORF. There is this undesirable attB site you find in the middle of the ORF and promoter, and it may be that it interferes with the expression dynamics. These are some points I would like you to consider when it comes to gateway cloning. So attP and B are involved in gateway. A similar ecosystem of cloning is called univector, which works like gateway but it relies on LOXP sites. So both of these are recombination-mediated cassette exchange cloning methods. There is an orthogonal approach that uses homologous recombination. Some methods in this space are infusion, magic, CEFC. Perhaps you will see them in a separate video, and maybe I will see you there as well.